Good afternoon, everyone, and welcome to this Genomic Forum last session on biodiversity. My name is Federica Di Palma. I'm the Chief Scientific Officer and Vice President of Sectors of Genome British Columbia. And indeed, throughout the day, we have seen some extremely engaging and informative sessions, and the common thread amongst all of them throughout the day was indeed biodiversity. Biodiversity, which is a critical and important component to human health, economies, and our livelihoods. The last UN Intergovernmental Panel on Climate Change report has recognized more than ever the interdependence of climate, ecosystems, and biodiversity. Biodiversity is being threatened by the current changes in climate and by our human activities, and these changes, unfortunately, are outpacing much faster the ability of these habitats and species to respond. Canada itself possesses a significant amount of biodiversity of species and ecosystem ranging from desert to Arctic. Many of these are at risk of disappearing. Many are being degraded and fragmented, particularly in our province of British Columbia. In this afternoon session, we are going to explore how genomic technology are already aiding ongoing efforts in this area, characterizing, preserving, and managing our biodiversity in an attempt to build resilience in our ecosystem, to adapt, to mitigate to these devastating changes. But also, we are going to discuss how such innovation really needs to integrate, stronger than ever, knowledge across the natural, the ecological, and the economic sciences. So to kick off this panel discussion, I'd like to introduce you Rachel Meyer, Dr. Rachel Meyer, who's an adjunct professor in Ecology and Evolutionary Biology Department at the University of California, Santa Cruz. She's also led much international work on science policy. Please welcome Rachel Meyer with a quick talk. Thank you.
Thank you so much,
Welcome,
Federica.
Rachel.
A pleasure to be here. So today I'm going to be telling you about a tool that we're using called environmental DNA to do biomonitoring, which includes a lot of biomonitoring to understand climate change in California. So what is environmental DNA? The roots of environmental DNA as a topic of study come from paleobiology and microbiology. For example, the ocean microbiome, the human gut microbiome, all of these rely on techniques like metabarcoding, where we need to understand new ways of tracking different species and strains in systems where we can't necessarily see everything with our eyes and take pictures and collect everything. Lake sediment cores, going back thousands of years, still contain some DNA, and really amazing paleoclimate studies have been done using sedimentary ancient DNA. And all of this is possible because of the amazing amount of sequence data that's been generated for different species around the world. And Canada is a leader of this with the Barcode of Life data system, now BioScan, where we started in 1982 finding common parts of the genome that all species have, where each species has its own unique barcode. So we can use that to track biodiversity over time and space now. So environmental DNA is the cells that lyse and when something dies, or it's the cells in the DNA that's in hair that gets shed, or your skin cells that shed, or when a leaf breaks apart, it degrades. And so all of this DNA that's in the environment can persist for quite a long time in the bottom of a lake, like the sediment example that I just shared. It can just be bound to particles, but you can also just store it in a freezer and store it in different storage buffers and keep it as a research resource for a long time. Some great examples are deep lakes. Even in warm areas, we're starting to discover, like in Hawaii here, that there's DNA that's well preserved. It's not just in the Arctic. and in other cold areas. And there's even DNA, environmental DNA, that you can get out of the air and out of the stratosphere. We're starting to understand how DNA particles travel really far distances around our planet, and even contain still living cells. And in museum collections, there are environmental DNA sequences in each of these collections that might tell you about the microbiome of the organism or the habitat that it was living in. But environmental DNA is also, as much as it preserves well, it's very ephemeral. It can be broken down by sun, it can be broken down by pH, heat, nucleases that are free in water or in soil. It can be eaten by microbes, and it can be bound to particles like soil. More cations will bind DNA, but that depends really on the soil type. So you might have some soils that will keep DNA for a long time and others that won't. So we can actually leverage these gradients of ephemerality to examine really recent events, or to examine seasonal changes, interannual variation, going into very deep timescales of decades to thousands of years. And this is important because a lot of people already say that because global temperature is so out of whack, that the current situation is not normal. So you can't even study systems in some sort of steady state or normal state. So with environmental DNA, people often use quantitative PCR to measure single species, or metabarcoding to measure the community composition of a group, say all bacteria. You can measure the relative abundance of dominant communities of species with metagenomics, and you can measure groups of target species, even very rare target species, with techniques like hybridization capture or multiplex qPCR, or you can try to get the entire tree of life with multi-locus metabarcoding. So how might we use all these measurements where we can track environmental DNA through space and time? Well, here's a classic example of finding the great crested newt in the UK. This is an endangered species where both metabarcoding and quantitative PCR are useful to be able to detect the great crested newt. And it's actually used in models to add tax to where you might want to build a home. And if you're building on great crested newt habitat, you would pay a higher tax, and that goes towards conservation of more land for the great crested newt, protecting endangered species against climate change. Here, this is maybe particularly a little hard to see, but this is the New Zealand mud snail. And quantitative PCR of environmental DNA is correlated with so the amount of DNA that you can detect is correlated with the density of the New Zealand mud snail in different study sites. So we're starting to see signs that it actually does reflect the abundance of animals in an ecosystem. And I love this example. This is one from Beth Shapiro, where environmental DNA, sedimentary DNA containing mammoth DNA, was used to track the extinction of mammoths on St. Paul Island. But also you had spores, so you could even think of tracking these spores with DNA. In this case, they were actually looking at the spores, but these are fungal spores that live on mammoth feces. And when the mammoths go extinct, you see this other indicator of this fungus, the dropping out. So we're starting to see these interspecies relationships. And what this told us was that the mammoths didn't go extinct on St. Paul Island because of something necessarily human caused, but they lost their fresh water, which was why that slide had water disappearing. And so now we're seeing that there are microbial proxies that might be really useful for tracking large animals or other species of interest. So if environmental DNA is so cool, why don't we see it used everywhere? Well, there's some major problems. We don't totally understand the temporal window of eDNA signals in different environments. We don't even understand the different signals in water versus sediment versus soil versus air. And we're still learning that some species don't shed very much DNA. We haven't sequenced everything on the planet. About half of the species in California have zero DNA sequence data in public databases. And we still haven't really figured out how to estimate the amount of DNA signal and how that relates to density or the number of individuals. So how can we close the gap? Well, we need more biodiversity sequencing. We need more pilot projects and case studies, and we need to get the research and management communities together to advance this tool. When we work together, we learn a lot from each other. Biology and biodiversity scientists in particular are actually the most collaborative science group. Often this happens at a bar, but we won't go there right now. We read each other's publications, we see presentations of our results at conferences, but oftentimes waiting for publications might put you 3 years behind the state of the field, because that's the average time from starting a project to when you actually are able to share your results with everyone. So how can we build collaborative networks that replace the bar, where you're talking about your new projects, your new ideas, and something that's ongoing or emerging? So I'm going to tell you about 3 examples of where we're trying to bring people together at this pre-publication stage to help advance environmental DNA. First is the Cali DNA Citizen Science Program that we started in 2017 at the University of California. This is a cross-campus project where lots of people come together. We have a Slack group, we have common methods, we do certain events like BioBlitzes, we have trainings, and we all, through using common methods, can compare our environmental DNA results and try to advance the questions that we can ask with this tool. There's too many people to name, but it's a large group and you're welcome to join the team. Our sampling design is where we collect little 2 ml tubes of soil or sediment so that they can be archived in freezers, or we collect water through filters, and then we save the DNA. We have lots of grassroots projects where we try to build a feedback loop of sharing results and with the community and trying to build new projects or maybe generate better sequence data and continuously improve environmental DNA research. Here's some examples of what the Cali DNA program looks like in action. And when we take samples back to the lab, we do multi-locus metabarcoding, trying to track all different major groups of life. Here's our first project from Pillar Point in California. It's an intertidal survey that has lots of iNaturalist observations. On the right, you'll see Pillar Point. There's 3 different segments of the beach. And if you compare the iNaturalist observations, which are housed, they're aggregated in GBIF, right, the Global Biodiversity Information Facility. If you compare the environmental DNA results and the GBIF results according to the class, right, kingdom, phylum, class, order, family, genus, species, you see that most of them overlap, but eDNA gets a lot more. But then as you start to refine your taxonomic scale, we see less and less overlap. Where at the species level, we're looking at an entirely different part of the ecosystem, where there's very little biodiversity, where we're seeing in common eDNA and GBIF data. But even if we don't have a lot of overlap, and we don't necessarily know if all of the taxa that we're detecting are true, we still get these incredible patterns because we're dealing with big data, thousands of species across hundreds of sites. And so we can actually make species interaction or co-occurrence networks. And we can find different locations that have similar communities using ordinations and other statistical analyses. We have a paper that came out last year in Ecological Applications where we were exploring landscape analyses, where we were relating remote sensing data and environmental DNA data. And we could actually take environmental eDNA and put it in a model and recapitulate a lot of these maps, like the EPA's map that describes the ecosystems of California that took decades to make. In the Center for Natural Lands Management, this is my hometown in the desert, there are 26,000 invasive crayfish in a small oasis, and none of them yielded enough DNA to detect through qPCR methods. So we turned to those microbial bioindicators similar to the mammoth paper that I described earlier. And using tank experiments, we were able to detect fungal and bacterial indicators that now add to the manager's toolbox to be able to detect invasive crayfish using a little bit of sediment from the Oasis. And now that they've eradicated the crayfish with poisons, we're actually using that as a means to track if the crayfish have returned, because those little buggers can hide. At the same time, we're also getting all of these interesting species that are very hard to detect with the naked eye, such as the brain-eating amoeba that's in the Oasis. Co-occurrence networks are super useful for understanding the functions of invasive species, as well as giving us another means to detect them. And we know that with climate change, invasive species are becoming an exacerbated problem. We have other projects such as on the Los Angeles River, which is super urban. Some of it is concretized on the bottom, so it's not a natural river anymore. And we can see with environmental DNA metabarcoding how some sites start to look very different from each other. And we also are finding ecological indicator species, which might tell us new ways of tracking environmental health. And there's a really lovely course that's been developed on environmental DNA at the University of California that you can check out called Biodiversity in the Age of Humans. So it's also a very good teaching tool. So in National Historic Parks, with the National Park Service, we're starting to do surveys of different streams where maybe they don't have very much biodiversity information, then they want more survey data. Sometimes the rocks can be very challenging and we don't get very much information from sediment, but we get a lot from water. We can see how streams are different from each other. I'm just giving you some quick vignettes here. We can see how over time and seasons, how communities shift. In Glacier Bay, we're starting a project looking at harmful algal blooms and how they affect the whole community around them, trying to understand tipping points. This is a collaboration also with the Hoona tribe in Alaska. And online now, we have about 4,000 registered users who are starting to engage with us using our protocols. We're also generating some reference DNA data for aquatic invasive species and fish and other endangered species to try to help advance the reference databases that we have. So with all this reference data that Canada's adding, that the whole world is adding, we need to be able to find more species in our existing eDNA data. We can reanalyze that and reanalyze that. And so people need more choices. We don't yet have standardization. So we're trying to make a space to work together called the eDNA Explorer. This is a new project, and I'm really happy to talk to people about their ideas of how to build a really useful Explorer tool. We want people to be able to co-analyze their results or their eDNA data together and share it pre-publication. We'll have events and places where you can promote your own work and your own collaborations. And hopefully we can get people to share lots of data. Because if you can share data, you can co-analyze it and learn from each other. But we need lots of policies that need to change in order for us to be able to safely share data. And we also need to recognize that one assay does not fit all. So we need to understand the differences in qPCR, metabarcoding, and all the different approaches people are taking to make this tool work. Thank you so much.
I'm muted. Okay, thank you very much, Rachel. That was fantastic, actually. Great introduction to start us off today. Thank you very, very much. I'd like to introduce to you right now the remaining members of our panel so that we can begin our discussion. Please welcome Beth Shapiro, who's a professor in the Ecology and Evolutionary Biology Department, affiliated to the Genomic Institute at the University of California, Santa Cruz, also a scientific rock star who has authored a couple of magnificent books. And I urge you to go and get the last copy of the last book. Also welcome Danielle Iñas, a very dear colleague of ours, assistant professor in in Indigenous Natural Sciences in the Department of Forest and Conservation Sciences at the University of British Columbia. Welcome, Danielle. Teal Brown-Zimrin, Managing Partner at Galvanize Partner. She has operated for many years in the private, public, and non-profit sectors. Thank you for joining us, Teal. And also Karen Helbing, Professor in the Biochemistry and Microbiology Department at the University of Victoria, and also one of the recipient of one of our large-scale applied research awards from Genome Canada in environmental DNA. Welcome, Karen. And last but not least, Steve Jones, Head of Bioinformatics and Co-Director of the Genome Science Center at the BC Cancer here in BC, and also a recipient of another of these large-scale applied research award from Genome Canada, leading the Canadian Biogenome Project. Welcome, everybody. So, Rachel, thank you so much for giving us a fantastic overview of what eDNA is and what we can do. So I'd like to really go straight to Karen and remain on that thematic for a little bit and ask, there are many points that Rachel brought up in her talk, including the lack of standardization and what should be done moving forward, and how can eDNA really be used for purposes of biodiversity monitoring. Karen, please tell us a little bit about what you are doing to aid such a standardization.
Thank you, Frederica, and thank you, Rachel, for giving such a wonderful talk to introduce this session. It was absolutely fantastic. What we're doing is we have a large-scale applied research project called iTrack that is geared towards addressing some of the issues that you so eloquently showed, Rachel, with regards to filling in gaps on the lack of genomics resources that are relevant to Canadians and others across the globe, as well as making the measuring eDNA accessible to people in terms of being able to use the techniques that are developed with confidence. And that involves also standardization of methodologies and so on, and also providing a framework for regulators and policymakers to make decisions about eDNA and how it can fit into management and decision-making structures. One of the things that we have done in Canada that we're very excited about is we have actually developed the first eDNA standard, national standard through the Canadian Standards Association on eDNA terminology and minimum reporting requirements, which involved a whole slew of people nationally and internationally. And we're also following up with a second eDNA standard on targeted assays for eDNA detection. So we're very excited about how things are moving forward, and we're very much looking forward to also developing a greater network internationally with colleagues all over. To coordinate genomics resources, and it's fantastic to see the wonderful progress that CalEDNA has done already, and we're very excited about the prospects.
Thank you, Karen. Thank you very much. And still remaining a little bit more on the theme of eDNA and just monitoring of biodiversity, given that we have talked, this session has really concentrated on the urgency of the climate issues. Do you, Rachel, Karen, or Beth, see a future for eDNA monitoring in responding directly to the mitigation of climate change? Having seen, particularly in the West, I gave a little introduction this morning. Having seen what has happened over the last 3 years, particularly in BC, but generally on the West Coast with the fires and the floods. Karen, yes, you had your hand up.
Just yeah, just quickly, in terms of being able to effectively respond to issues around climate change, one of the cool things about eDNA is that it is very conducive to very quick responses, provided that one has the appropriate tools and genomics resources available at one's fingertips. Then you can actually be way more nimble in terms of identifying and monitoring change as it's happening, rather than being dependent on many difficult and conventional methods for doing environmental surveys. I'm curious about how Beth and Rachel feel about that.
Beth.
I just speaking to the idea of urgency, we're seeing that the climate is changing really rapidly. And this summer in particular is supposed to be, in California anyway, this 1,200-year drought. We have all of these, the horrible things that are happening right now. And I think, as Karen has said, and as Rachel pointed out, this is a really powerful approach to being able to catalog biodiversity over time, but we need to know what the baseline is. So we need to be able to get out there now and collect these samples. We don't necessarily have to process them now, but we have to have these samples at our fingertips so that when the climate changes, when there's been a fire, when there's been a drought, we can go back out to these same places, extract the DNA and characterize it, and then compare what's happened. And because there's so many things we don't understand. How does seasonality impact DNA survival and persistence? How does succession impact DNA survival and persistence? There's a lot out there that we don't know. There is a great urgency of getting out there and just collecting these samples so that we have them for work that we intend to do in the future, because this is a powerful approach now. And as the technology becomes more sophisticated, more specific, it will become even more so.
Thank you, Beth. Thank you, Beth. I think that I've heard from Rachel, I've heard it from Karen, I've heard it from Beth. More sequencing is needed, and I really liked the concept of a baseline right now. And that's what we need to do in order to really accelerate the urgency of our response. Okay, so I'm quickly moving away from eDNA right now, because I want to go around this panel and a little bit on a tour of the different sort of innovation technology that have been used to really characterize biodiversity. And I'd like to go to Steve and ask Steve is using a different approach to biodiversity, is using genome sequencing. Steve, tell us a little bit about what you are doing and how you intending to contribute to this.
Sure, yes. Yeah, I mean, we can step back a little bit and we can just really think about, what are the basic questions that we need to understand, about the animal and plants that surround us. And we really don't have a good idea on how all these animal and plants actually work. And that really, is the mainstay of the goal from the Canadian Biogenome Project and its umbrella organization, the Earth Biogenome Project, is to start to understand much more about the genetic makeup of these species. Canada itself has 80,000 plants and animals that are multicellular in nature. Canada is the second largest country in the world, and it also has a very large interface with the Arctic. So this really, many of these plants and animals are going to be the ones that are going to be affected quite severely by climate change. So one question that we're really looking at is, how do these animals work? How do these plants work? And really by looking at the genomics, getting a high quality reference genome for many of these, and conceivably the goal ultimately would be to get a high quality reference genome for all of them. We can start to understand the different proteins and evolution that has taken place within these. What's actually really exciting now is that over the last 24 months, we've seen the kind of the invention, I guess, of a technique from Google called AlphaFold, which can predict computationally, what the three-dimensional structures of the proteins would look like from their primary sequence. Now, of course, there's some error bars, in those structures, but presumably the technology is only going to get better. But it's really fascinating now that we can go from sequencing a genome, which at this point is actually very, very routine, not trivial, but routine, and conceivably in a few weeks get a reference genome, and then conceivably within a few more days or weeks get a three-dimensional structure prediction for every one of those proteins. So we've got the parts list of an organism, and now we've got what that part actually looks like. So it's a really exciting area to be looking at how we can address, to me at least, a basic fundamental understanding of the plant and animal diversity that actually exists within Canada.
And in your effort, Karen wants to make a comment. I'll just say that in your effort, Steve, you can then address it later. In your effort of doing so, I think one of not only the genetic information that is, of course, released, as you say, through this effort, which is going to be pivotal for years to come, but there is that first effort of vouchering samples. You also, I believe, work very closely with museums, with organizations for doing so. I think this is often an underestimated portion of really cataloging and preserving our biodiversity.
Yeah. I mean, that's a large part of the effort, is to make sure that what we're sequencing is exactly what we think we're sequencing. Clearly, that's important. It's important that we understand the provenance of those samples, so we understand that they were collected legally. Clearly, that's important as well. And really, just being able to preserve as much of the sample as possible. It's quite an esoteric term, vouchering, but really it's about kind of making sure that we're making the most of our understanding of the sample that we use for genomic sequencing, and that we're storing as much of those samples as possible for future generations. For larger animals, we can only, for instance, store or maintain maybe a sampling of tissues and organs. For some species, by the time we've extracted the DNA, there's nothing left except residual DNA, but we can still kind of voucher that and store that as well.
Thank you. Karen.
Well, one of the really neat things is that even like what Steve is doing is like massive, amazing assemblies of many organisms. But we have the voucher specimens are very important, of course, to have properly identified. But when we have situations where the DNA is degraded from these valuable specimens, making full assemblies of entire genomes isn't possible. But one of the really neat things is that what we can do is we can take portions of those genomes and actually assemble mitochondrial genomes more readily with these degraded samples. And one of the things that the iTrack DNA project is doing is developing methodologies for being able to take degraded samples and still get full mitochondrial genome assemblies out of them, which are much smaller, but can be used in many of these important applications, for example, in environmental DNA applications.
Thank you, Karen. And Rachel, please.
Just really quickly want to say that environmental DNA does not just have to be to track taxa, to track species. We could track genes. We could understand the functions, what the environment is feeling, the stresses that it's feeling by looking at genes. And we can do that if we have full genomes for everything. So I'm so excited about the Earth Biogenome Project and what you're doing with Genome Canada. It's great, and it's going to be a revolution for eDNA. That's all.
It is indeed a revolution. Thank you very much. And I'd just like to move quickly to Beth. And Beth, you are the head of a paleogenomic lab. So degraded DNA must be something that you deal with on regular basis. I wondered whether you had any thoughts about that following up from Karen, but also I wondered whether you could tell us a little bit more about what opportunities are there for genomic editing, for gene editing in this space.
So this is two different questions. Thinking
Two
about
different
paleo,
questions.
thinking about paleo stuff, we're accustomed to working with really garbage quality samples with tiny little pieces of DNA, and it's always surprising how much really excellent and useful information we can get out of these mostly degraded samples. This is because after an organism dies, the DNA that's in its cells starts to get broken down by things like UV light from the sun, or freezing and thawing, or microbial action just getting in there and chewing stuff up. And then over time, the DNA gets broken down to such an extent that we can't use it anymore. We can't identify the pieces too small to allow us to accurately identify what organism it came from. Paleo stuff has actually been important in eDNA really from the early stages of the field, where one can, for example, go into a permafrost region in Yukon, which is where I do a lot of my field work, or take a sample from a core from the inside of a lake, like we did with the Mammoth Project. And with that, you can actually track changes in the community composition over time by extracting DNA from these different layers and then tracing how these communities and organisms are either the communities are evolving, or even perhaps the organisms and genes are evolving. So that paleo perspective has been really useful. What's challenging about it, and this gets back to the urgency that I spoke to earlier, is that the time scale represented by one of our little samples isn't 1 year or one season, as it might be if we're going to go out and collect surface sediment samples right now. Instead, it's averaged across some number of years, which depends on the sedimentation rate, the rate at which the sediment and the soils and the DNA is collected in that lake. And so we can't get as precise an estimate, temporally precise an estimate of what's going on, how these communities are changing. But we can see the big picture, how tree line moves as glaciers advance and retreat, whether you have community perturbations that are associated with an extinction event, etc. So that's my plug for paleo stuff in eDNA, or for using paleo perspectives to try to understand how ecosystems are changing over time. It's all very interesting and fun, and also the past by its very nature is different than anything that exists today, and so is absolutely ripe for discovery. We've discovered new species of equids, 2 of them, in fact, working in the Yukon for over the last couple of decades. So we had no idea we were looking for these things, but we discovered them. So it's fun, it's exciting. It tells us about how biodiversity changes over time. And I think the reason that you then asked me about gene editing is because all of my work in ancient DNA means that whenever I am spoken to by somebody from the media, what they really want to know is whether it's possible to bring a mammoth back to life. And so I've gotten very involved in the potential of using this sort of biotechnology, what that one would need for de-extinction or bringing a species back to life. And I've done a lot of thinking and speaking recently about how these technologies are incredibly cool and useful for potentially helping living species to be able to adapt to their rapidly changing environments. And I do think there's a lot of potential there, but that's a really big can of worms to open, and I'm not sure you really want to get there.
It is a big can of worms, but I actually want to open it a little bit. Because I think this is our opportunity as we are engaging with the public in this genomic forum to really have the public think and understand what we scientists that work in this area with the genomics mean by de-extinction. Because let's face it, Beth, we are not going to recreate a mammoth. So what do you really mean when we talk about de-extinction and the potential to use this ancient DNA? I really like to explain this a little bit more.
Well, I do think when people say de-extinction, they are thinking about bringing extinct species back to life. But what I often say is de-extinction is not possible. Extinction is forever. We're never going to get a 100% identical copy of something that used to be alive. But these technologies could be useful for resurrecting extinct traits. For example, maybe we want to just riffing a bit on mammoths, maybe we want to take an Asian elephant and make it cold adapted. Well, can we learn what it is about a mammoth that distinguishes it from a tropically adapted Asian elephant, that we might be able to use these technologies to identify the genes underlying these traits and then transfer these genes to developing embryos using gene editing technologies, and then use cloning, or maybe we can transfer it to cells and transform those into embryos and use cloning to bring this back to life. Here's an example. So the black-footed ferret used to be really common across the North American plains, and then it nearly went extinct. In fact, it was believed to have been extinct until the last remaining population of black-footed ferrets, a remaining living population of black-footed ferrets, was discovered outside of Meeteetse, Wyoming. So there was an all-out effort launched to figure out how to captively breed these individuals, and it worked. But there's a problem, and that is that when these captively bred black-footed ferrets are released into the wild, they eat a prairie dog. That prairie dog is often infected by plague. The black-footed ferret gets plague and it dies. So you could vaccinate them, and then you can capture them and revaccinate them, but this is not a very good scenario for long-term conservation of the species. But there is another way, using these same tools of genome editing and engineering that one would need for de-extinction to actually work. The evolutionary cousin of the black-footed ferret, the domestic ferret, evolved alongside plane in Eurasia and is naturally immune. So if scientists could discover what it is that makes them immune, and no one knows this yet, but there's a team from San Diego Zoo, U.S. Fish and Wildlife, and a nonprofit called Revive and Restore that are working on this, we discover what it is that makes them resistant. We could use these same tools, insert that gene or those genes that evolved in domestic ferrets into a black-footed ferret, and then clone them using the same technology that brought us Dolly the sheep, and that brought us last year a black-footed ferret clone named Elizabeth Ann, who was cloned from 40-year-old preserved cells of another black-footed ferret, right? So we know we can clone black-footed ferrets, and then transfer this trait between species so that we could create a black-footed ferret that could survive in its environment of today, which includes plague. So this is an example of how we might use these technologies. Imagine being able to understand why some corals can survive in warmer climates, or some spruce trees are resistant to bark beetle attack. If we could understand what the genomic underpinning of these resistance traits are, we could transfer them between species using these technologies, using biotech to actually help these species to survive in the rapidly changing world of today.
And thank you so much, Beth, for having used an example which is so wonderfully has described ecosystem interactions, the complexity of those interactions, or how we can actually use a genomic sequence for future innovation in conservation effort, but also building resilience within a species and perhaps within an ecosystem. And that's why, and in the same time, we've already explored how we can monitor and collect a baseline with techniques such as eDNA, and we have seen, because of this genetic editing that you have just explained to us, how collecting and cataloging the full genome sequence in an effort like the one that Steve is doing is absolutely pivotal. So thank you. You just have an incredible panel. So now I'd like to move on from this, and I really would like to talk a little bit to Danielle and to Teal, and Danielle, you are an enrolled member of the Cordarline tribe, and you have been doing a lot of work on how global change, so climate change and landscape disturbance, is really impacting ecosystem function and indigenous communities. We've heard from Rachel's talk, we've heard a little bit from what Beth has told us, and from Karen, and Steve hasn't told us, but I do know that the Canadian Biogenome also has a very important connections to this. Tell us a little bit about what you do, why is it so important, how is it impacting indigenous community, and what is the opportunity for this genomic technology to engage this community and for us to learn from moving forward. Please.
Yeah, thank you for the introduction, and Rachel for your great anchor talk there. And this is a fabulous panel, and there's so much to talk about. I think there are so many overlapping issues here, and so we'll see how far we can get with this. But in terms of Indigenous communities, why do I study what I am studying, and why is this important for a variety or all Indigenous communities, is that because that Indigenous communities are stewards of land and water and forests, and there's an incredible biodiversity and ecosystem services and health impacts that should be considered here. And in a sense that if we can protect and strengthen the rights of Indigenous communities, and as well as respect the Indigenous knowledge, then that will help us in solving some of these climate emergency issues. And so not even just from a biodiversity standpoint, which is very important in and of itself, but also there's a lot of different kinds of diversity that needs to be protected, not only knowledge, but how knowledge is transferred to the public as well as different communities. And also there's a great deal of cultural importance here. And as we think about intergenerational knowledge that gets passed, and the relationships within ecosystems, and how these Indigenous communities are fabulous stewards of ecosystems, not only from a health perspective, but just from passing down the knowledge that they've always had. And so this is absolutely critical. We're at a really important time, and Beth touched on this a little bit in terms of understanding biodiversity and how important it is to have kind of the pre and post event in terms of understanding what are the implications of any human-caused actions and their impacts on biodiversity and ecosystem health. Can we have or can we maximize the information before something happens? And of course, now we're at a time where everything's happening much faster than we had predicted, and the outcomes are absolutely devastating. And so now we are at an opportunity here. If we not only want to protect and strengthen Indigenous communities and their rights and their knowledge, well, how do we move forward with that? How do we interact with these communities? And all the other panelists have touched on this a little bit in terms of, and especially Rachel in her talk, and talking about, well, there's maybe not one assay that fits all. And in the same way that when we interact with communities, especially Indigenous communities and communities of color, that there is no one template that will tell us exactly how to do this with each one of those communities. And so, yes, there is some nuance to that, but what that means is that there needs to be some relationship building, building of trust, in terms of this kind of data collection. There's been harm in the past, to various communities. And so there really is an issue of trying to build trust if we want to move forward. And it's been touched on a little bit as well, that thinking about changes in policy. So if we don't have a one size fits all, well, how do we have, in a sense, a way of relating to these communities and interacting with these communities, and have policies that are in place to ensure that their knowledge is protected, their rights, and the information that can be collected while on the land of these Indigenous communities is protected as well. So there's certainly so many things to talk about here, and I'm happy to answer any follow-up questions to that.
Thank you, Danielle. Steve, why don't you tell us a little bit what the Canadian Biogenome Project is doing to this end?
Yes. Well, what we're doing is, and we certainly haven't outlined any specific plans, because that's really what the engagement with Indigenous communities is going to do. We're certainly not prescriptive. But what we've noticed, of course, is that many of the plants and animals that we may be sampling or wanting to sequence may have cultural significance for groups, may have economic significance. And so we just want to make sure through consultation how we might work with Indigenous communities, approach these species, and really see how Indigenous communities might want to be involved in the understanding of those species. And certainly, I think there's a lot of interest in understanding how the genomics of these species may help preserve them for future generations.
Danielle, do you have any consideration or advice in terms of how in communities should be engaged in this type of technology? In technology, we've heard about eDNA monitoring. We've heard about what Rachel has told us. There's an immense opportunity for engagement. So, yes. And we've heard
right, yeah.
I wanted to make wanted to make just another comment that in the agri-food session today, earlier on, we've also heard that as we move towards a more sustainable type of attitude, that there is knowledge we do not have that may indeed help us, and that comes from these communities. So this is quite important. Yeah.
Yeah, thank you. I think there's a few things I think that can help. And one is to communicate early and communicate often. And so in a sense, this is really about transparency. And again, like I mentioned, about building that trust. And so I think part of it is comes down to science communication in a sense. Are we doing a good job of communicating and being transparent about what kind of data collection is going to happen, what will happen with that data that is collected? And on the flip side of that, if this is collected on Indigenous lands, well, have those communities been consulted? And we have to think about data sovereignty here and make sure that, again, these communities, their rights and knowledge are respected and protected here. And so this does really involve a lot of talking. And so that's why I speak to the idea of engaging early, engaging often, being very transparent, and even when that has been established, to make sure that what has been collected, any information or conclusions made for that, is in consultation with those Indigenous communities. And again, this does come down to protecting knowledge and culture and rights here. And so there is definitely another issue of access. And access we can think of in a few different ways. One is open access to data, and I'm sure this is something we've all on this panel have thought about, either in terms of the benefits of open access, and as well as some of the cons to that. And this is an instance where involving these Indigenous communities is going to be incredibly important. As I mentioned, no one size fits all, and I think we recognize that as a group here. Yeah, so it's a tricky balance here, but I think if there is early involvement in true collaboration and co-partnering with these Indigenous communities and being transparent, I think there's a good way to move forward.
Yeah, you touch upon so many good points, the data points. I mean, we could, by any means, we are talking about everything to do with biodiversity. There's a few things that we won't be able to chat about or even go deep in, and certainly data is one. But we are certainly very conscious that human health has brought us the attention of the equity and diversity of data and the ownership of data. But of course, the environmental context of genomics has the same issues. We are talking about data that are really important for the livelihoods of this community, that come from the lands where this community have resided for a very long time. They have a deep knowledge of this. And there is a community I've interacted with, and I will never forget what the leader of the tribe said once when we asked the question, what would you like from us doing this type of work? And I think he said, biodiversity is everything to us, and it's our culture, it's our life, it's everything. It's very intimate. It's an intimate concept for us. You know everything about it. You know everything that is in our land. We do not know anything of what you do with it or how you do with it or do not even understand it. And I'm just going to leave this topic here because with this, but those are words that I'm never going to forget. And they are obviously very always thinking about. So switching gear a little bit, I like to talk to you, Till, because what was great about this panel is there's a common thread about the genomic technology and the biodiversity, but the interdisciplinarity of us as well. It's really fantastic. And I wanted to ask you, from your vantage point, really, what your perspective really is on these issues, but also what role market-based approaches may actually play here. Is there a role for scaling up these technologies, and and how can we actually, yes, harness the power of the markets to deliver some meaningful outcomes for biodiversity?
Thank you so much, and thank you to each of you for this incredible knowledge. Danielle, I was just furiously taking notes on a few things you were saying, and I'll reference them as well, because I think one thing to kind of level set, and we probably all know this here on the size of these markets and the potential, is that for example, BCG estimates that something like $23 trillion, 30% of global GDP, could be disrupted by genomics. So the potential for this science to change the way we do everything, the way we make our clothes and wear them, the way we eat proteins, the way we create energy, is obviously so substantial. But how do we ensure that within that, we are carving out a specific piece of that massive biorevolution that is focused on climate and is focused on biodiversity and is focused on our natural landscapes? And I think, Danielle, you pointed to this. It's really in the how. How are we going to use markets, yes, to deliver some amount of private sector discipline, a look to scale and therefore impact, but also to do this in a way where we are listening to land stewards, understanding what is wanted and what is needed, developing the IP that can deliver that, the scientific advancements that can deliver those solutions, and doing it in a way where the whole ecosystem is together in the beginning so that we are incrementally de-risking the potential of biotech solutions that can deliver carbon removal and core resilience of ecosystem services. So I'll put a finer point on that. Think about, just broadly speaking, assisted adaptation. There are carbon-dense species like mangroves, like kelp. As we march toward a higher carbon future, Federica, you referenced this in the beginning, we will see novel pathogens and novel competition confronting these species as they migrate. There is every possibility to use genetic science, to use genomics to bolster these species' ability to thrive as they confront those novel pathogens and novel species, but doing that absent a plan for who wants this science, who wants this innovation, how will it be implemented, what are the partners and the land stewards and the organizations that will help us move this into some appropriate implementation that includes bioethics, the socio-political landscape, the cultural landscape. So I just think that part of the how, of the listening while we are developing the science, is so critical. And that's something we are doing and really thinking about a lot. I also want to underscore that while markets deliver discipline and the potential for scale, at the end of the day, there are market failures. The biggest market failure, perhaps, of human history is the failure to value biodiversity and to appropriately value carbon. And as a result, markets ultimately won't solve these problems. That is a policy problem. But markets can enable us, I think, to tell stories. We can carve out a piece of the biorevolution growth, so to speak, to tell stories. And I think a good example of that is the American chestnut tree. And in many ways, it's by virtue of the Cherokee Nation that tree went through the scientific process that it did to become resilient to an invasive pathogen using genomic science, and is now planted widely across the eastern seaboard of the United States outside of USDA regulation. So we told a story working with a group of people who were land stewards, who knew their land better than anyone else, who had a cultural and environmental significance for that species. The science, when completed, already had a pathway into implementation, and now the policy window has opened and the science was ready to scale. So that's what we're thinking about in the work that we do, is how to tell those stories, incrementally de-risk these markets, and then use the markets to deliver scale for these solutions, but to do the whole thing in partnership with the right people at the table from the beginning. Danielle, to your point, the land stewards themselves, the people who know their land better than anyone else.
Thank you very much, Teal. This is so important. So really underscoring the importance of bringing all the stakeholders at the table, that we no longer can get away with bringing the scientists around the table. The scientific knowledge, while foundational, it's not enough anymore. And this is quite clear. I thank you for mentioning the failure in valuing biodiversity. This is being enhanced on mine. In this panel, we did not have an opportunity to go more into this, but I do feel that indeed this is a part of the puzzle, because we have the GDPs which do not take into consideration our natural capital, and so different type of accounting also need to happen in order for the market to really unlock. And this brings me also to ask you any advice, guidance, thoughts around. We need investments in this area. I'm an expert in genomics. I also work with government in policy, but I often hear genomics has been around now for a while, for a long time. We've had the human genome, we've seen all of that, but I often hear it's something of the past. Genome sequencing is something of the past. This panel has highlighted how actually the transition for genomics into climate change is now. It's needed now, and it's needed, at the most basic and complex levels. So we're not really past, the characterization, and certainly we've talked about future innovation. So how do we convince our environment to make investments? Because this is not an easy question, Teal, but I'd like to have some thoughts on this.
Federica, just to make sure that I'm clear, the question is the narrative that will encourage capital to arrive in service?
Yes.
Okay. Well, that's a super difficult question. I think that there's an answer to it, but I want to acknowledge that that's just my perspective. I think that the answer is talking about the profound potential that these technologies have to capture incremental value through things like drop-in replacements of existing products. What are we already doing that we can do better using these technologies with substantial co-benefits and climate outcomes? New market opportunities. Where can land stewards gain carbon value that they're not capturing right now as we increasingly price carbon? There's complexities around that, and we can talk about that, but it is nonetheless a viable, real dollars, real prices market. So how can we deliver incremental innovation that has important and profound impacts for biodiversity, but allows land stewards themselves to capture the carbon value of their land? And then I think the kind of third place that I would think about is co-benefits. So if we, for example, produced a soil amendment that could deliver a water density or carbon density to an arid soil, you would see people that are managing that land for agriculture, smallholder farming, forestry, whatever they're using it for, they would gain the benefit of more water in the land. That would decrease the costs of water. So I think we have to look to economics to tell us where we're spending money today, replace those existing financial streams with ones that we think are beneficial for the planet, and use those to, again, kind of tell stories. At the same time, we have to push hard on the policy windows. We have to push hard so that these technologies are ready. And we are investing today because of those incremental co-benefits. But ultimately, what we're doing is we're getting ready. We're getting ready for the time when policy windows open enough for us to scale those technologies through market mechanisms that do value biodiversity or do value carbon appropriately. And that may come all too soon in ways we might not imagine by virtue of unfortunately, catastrophic events like megafire. So, we start in the places where there is remediation to be done and where there are co-benefits to be gained. And that's where we incrementally optimize, I think. And I do think that's probably a narrative for investment that looks at, offsetting existing costs as a first pathway in, most likely. That's probably too wonky for you.
Thank you very much. I have a question for Rachel and for anybody else, in fact, who would like to contribute to this. But what is so we talked about many things: support from indigenous local communities, but there's also the support of the public that is so important to bring this forward. All these concepts that we talked about today. So, what is the role? How can genomics and you've touched upon this in your talk, Rachel, help us really engage the public sector?
So the public really need to understand genomics now, because it's data that's going to help them understand when environmental conflict is happening and when they need to react. They need to move or they need to change their practices, because otherwise you have human suffering. That's the entire reason I started studying eDNA in the first place, was because to even predict where conflict is going to happen, you need models that are accurate, and we don't have biodiversity data in most of those models. So we're losing out on an opportunity to make them more than, say, 85% accurate. If the public can understand biodiversity data, genomic data, models, they can survive without suffering. They can make changes in their lives that are going to be good for the planet, be good for themselves. So I just think it's really important. And with environmental DNA, when you have so much biodiversity that you're considering, it's really easy for scientists to just ask the same questions over and over again that we're used to asking, because that's all that we know. And this is really complex. Biodiversity is extremely complex. And so we need more creativity, more different perspectives, other expertise to be at the table so that we can make sense of these patterns and processes that we're observing. So I think it can advance the science a lot and help protect humans from getting really hurt in the future.
I see a lot of nodding, Rachel. Okay, so for the last 10 minutes of this panel, if that's okay with you, I'd like to engage the public who has been posting questions while we were all talking. And so I'm going to read out some questions. I'll start with a very specific one for Rachel. Thanks for a great talk, Dr. Mayer. These are some really interesting projects. Out of curiosity, which platforms are you tending to use for the multi-locus eDNA work?
Wow, okay, thank you. Well, we had to build our own software because at the time there weren't software platforms that we were able to use for multi-locus metabarcoding that were efficient and up to the standards that we wanted. But now everything's kind of just as good. So we use Anacapa as a software, and then we put the data on our CalEDNA website. Even if it's raw, even if there are some errors, we kind of accept that with some, here's the uncertainty, understand that this isn't perfect yet, but evaluate it, give us feedback. And then we publish on NCBI, which then, goes to the global biodiversity data, genomic data bank and genetic data bank.
So for Karen and for, I guess, for Rachel, for whoever wants to chip in, are you training citizen scientists on the eDNA monitoring techniques? And how do you ensure that data standards and harmonization are kept?
Sure, I'll give that a go. I think it's absolutely essential, just to echo what Rachel was saying, how important it is to really to make things accessible to people. Demystify very complex science, but bring it really to the citizen science. Environmental DNA, as an example, is a really very highly conducive to certain applications with citizen science involvement, and putting together training modules for how to collect appropriate samples, and then how to curate it, and then how to analyze it. All of those modules involve different groups of people, or they can involve a continuum of people. So having all of those things accessible to the folks that want to use them is super important. So, for example, we're developing modules that are purpose built. And I know that others are doing similar things as well, where like training people to citizen scientists to go out and get samples. Requires a bit of standardization and so on, and that's dependent upon the types of samples that are being obtained, and where they're being taken from, and so on. And those are amazing opportunities to not only train people to do it well, but to engage the public, get them excited, get the kids out there, get the adults out there, and do a family thing, but do it in a scientifically rigorous way. It's not impossible. It's challenging sometimes to figure out how best to do it, but it's well worth it, and is, I think, the biggest opening for really opening up that dialogue, and also accentuating the relevance of what it is that people need to do in order to really learn about biodiversity and to do something to protect it.
Thank you, Karen. There's another fantastic question with regard to the citizen science. How can we best mobilize citizen science groups like BC string keepers and bird watchers to bring in additional eDNA data? Rachel, you're nodding. Are you doing this?
Yeah, we're in this renaissance where naturalism is cool again, and it's awesome. There's so many groups of people that are mobilizing now, and we need you all at the table. We need you to keep contributing data and doing analyses and being experts. So the more that eDNA researchers can collaborate with those community science groups, the more we can understand the biodiversity data that we get. For example, I could detect ghost shrimp in a sample and think, oh, this is weird. It's not normally supposed to be here in this month. And then someone says, oh, that's the fishing bait that we use. This guy down the street sells it. So you are the experts of your local environment, and we need you to keep learning and telling us what you're seeing. And, if you see something, say something.
So eDNA is very popular. So there's yet another question I'd like to address for the eDNA experts. In a lot of biodiversity and other ecological research, voucher specimens are vital to ensure replicability, etc. With eDNA, there is often no chance to collect traditional vouchers. What are your thoughts on dealing with this issue? Did we understand the question? I'm not sure I do, but maybe you guys do.
There's
Karen,
no
would you like to
voucher specimens that I know of for eDNA and national collections in the US. Does Canada have them?
You mean with respect, just like voucher specimens, or
environmental vouchers, like what Beth was talking about with collecting before the fire season, so that we can look for afterwards what happened.
Yeah. There's no national repository for that. So there's definitely a need for the community to build up a resource like that. Right now, there's a lot of smaller groups here and there that are involved. So bringing people together is going to be very important.
Hello, Dr. Shapiro. What species do you think will be targeted for the next Revive and Restore project? Also, I really enjoyed your book.
Thank you for the compliment. That's very nice. How do you mean the second book? Because I enjoyed that one a lot more than I did the first one, which was just about why it's not possible to bring mammoths back to life. Species targeted for revive and restore. So, I mean, they have a lot of projects that are ongoing right now. They're interested in Przeworski's horses and corals, and they have a really fantastic program where they've engaged with communities from around the world to create genomic resources that are going to be useful for conservation. The normal question that I'm asked is which species is going to be first to be brought back to life, and my answer, I will just reiterate, is that anything that is extinct forever. Extinction is forever. There will never be anything that is extinct that will be brought back as 100% identical copy in every way to the species that used to be there. So I really do love the idea of using and developing these technologies to try to protect species that are alive today, to prevent additional extinctions from happening, and hope that this is where all these engagements are going to go. But there's tons of stuff going on, right? There's the new company called Colossal that's just been formed that's taken over the Mammoth de-extinction project, and they're going to learn a lot about how hard this is and whether it's really possible, how far it's possible to go. I think this is really exciting because one of the things that those of us working in this field are often accused of is taking money away from conservation programs that are more tried and true. And in this case, this is clearly not that. They just went through a Series A fundraising round and they raised something like $80 million or something crazy like that, that is come from biotech. And this is new investment into technologies that will directly affect conservation. And this is exactly what we want. It's absolutely brilliant that this is happening now when it is needed. And I'm excited to see where all this goes.
Thank you very much. The last eDNA question, whoever would like to take it. Can we use eDNA to help address One Health-related questions, such as monitoring pathogens outbreaks? I have two nods, so take you, Karen.
Short answer, yeah, for sure.
I saw lots of hands up. Beth, Rachel, last comment on this.
One of the things that we're really interested in trying to figure out how to do is, it's kind of hard sometimes to detect pathogen in the environment. Maybe we don't even know what pathogen we're looking for. But what it might be possible to do, what we're hoping it's possible to do with eDNA, is especially if we have a time series of data or we have a spatial series of data, can we use the tools, the computational tools associated with eDNA data to understand what an environmental steady state might look like? And in doing that, can we then see some perturbation away from this steady state as a way of saying something is going on here? The environment is telling us that it's sick, that there's something weird happening, and then use other tools to dig into that to figure out what's going on. So I think there's a lot of potential in this space. It's certainly a space that needs development, but it's an exciting avenue for research that hopefully we're all going to be more involved with as we go forward.
Thank you. Thank you very much. So you have been an absolutely wonderful panel. I couldn't have hoped for a better discussion, frankly. So thank you very, very much for your time. And I really hope that there is some interaction amongst all of you, given all of the alignment and complementarity to what we have discussed. And thank you very much. And we can perhaps say goodbye. Have a great evening.